Hello.
Hi Serena, how are you?
Great. I'm watching the movies and the supplementary material. They're amazing.
Oh wow, that's great. Well, I also have a PowerPoint version, but it's way too big, I think, for people to access it. Hi John, you want to come up?
Hey, John.
Good afternoon. I'm in multitasking listening mode, but I didn't want to ignore you.
Well, this paper really amazed me. I'm glad Dr. Rupprecht can come and present it to us. It seems really groundbreaking. The supplemental videos of the paper are amazing too. Definitely recommend checking those out.
Could you repost the original paper?
Oh, yeah.
Yeah, I
Let's.
just added in the chat. Feel free to check it out. It's a bioarchive, so you can just access it. Everyone can access it.
The slides look really good too. Of course, I'm going to be biased and excited about astrocytes at any rate.
Hi Leanne. Hi everyone. We will start in around 7 minutes, so thank you for coming. I hope my background noise is not too bad. Hi Leanne, how are you?
I'm great. I'm looking forward to this.
Yeah, this is going to be a good one.
Serena, I haven't seen you for a while. How are you doing?
I'm doing really well. How are you?
I'm good. Just enjoying the last week before the start of classes. Just going for a little walk
Oh,
along the
wow.
ocean and looking forward to the talk.
Get some beach time, some astrocyte time. It's all good. I've seen a few really interesting papers about astrocytes lately. One came out in Nature just like last week, in a similar direction about establishing astrocytes as computational units. It's Very welcome trend.
Hey, Dr. Olu, do you want to come up to speak? Feel free to come. Hi, Peter. How are you? I hope everything is good. Thank you for coming.
Hi, Katharina. Nice to hear you.
Hello, Peter. So
Hello.
glad you could make it today. This is going to be fascinating talk. I'm
Thank
sure.
you. Thanks
We
for
will
inviting
start.
me.
Oh, yeah, of course. Thanks for coming. It's a great honor having you. This is such a interesting topic. Serena is an absolute fan. So
Definitely. I've just been well, I won't even say borderline obsession, but I've been obsessed with digging into astrocytes and their function for the past few months, and I saw this preprint in conjunction with several other papers on astrocytes and their role in actual computation. So I'm really excited to hear what what you have to say today, and I'm sure we'll have a lively Q&A.
That's good. I think that there are many recent papers on astrocytes and their role for computations. I think we can also discuss some other papers as well, if you're interested in the aftermath of the discussion of this paper.
Well, certainly. Let me follow you. The videos in the supplementary material, I was just telling the audience earlier, they're really amazing that you brought all that data together in coherence and
Yes, I agree. I would also say that the supplementary movies are maybe the most interesting part for newcomers because they really show how it looks like to do the experiments and how the data look like.
yeah, and just staring at the movies, it's just like, are we looking at what this little mouse is thinking of at the time? How do we understand what component is, what's really being represented? So, no, it's fascinating stuff.
Yeah, also at the same time, it was a big challenge. I started imaging neurons when I was doing my PhD. And in the beginning, I always asked myself, okay, I see some blinking cells, but what does it mean? And it's really difficult to relate it to some cognitive stuff. And of course, we can do this partially, but there's so much unexplained variability which don't know how to explain it. I think that's really interesting and unexplored.
Yeah, you can really get caught up in just trying to interpret it, and but it's so compelling.
Yes, I think this what I'm presenting now, in the following hour, is maybe one aspect of astrocytes, which is, I think, very central, but there are many other activity patterns of astrocytes which are not described by this approach, but which would be described by other approaches. So I think it's just maybe the first order description of what astrocytes are doing.
Mm-hmm. Yes, definitely.
I think we can slowly start, right? So people will keep dropping by, but let's just start with the introduction slowly and then we go from there. So, welcome everyone to Science Society, and of course, a special welcome to Dr. Peter Rupprecht, um, who will be talking about his research here today. And before we start with that, let me give you a little bit of information about our speaker here today. Peter grew up in Germany, and he's a second son of a carpenter. and a kindergarten teacher. He, after he did his high school degree, he did his civil service. So I think it changed the last years, but back then in Germany, you, as a male, you had to either go to military service or you could do civil service instead going to the military. In a hospital, he did that in a hospital, and after that he studied physics in Bayreuth, Germany, and Lyon, France. After his master's thesis in 2014, he started his PhD in neuroscience with zebrafish at the Friedrich Miescher Institute in Basel. And he, for his PhD, he won an award, the Ruth Shiki Originality Prize, for thinking out of the box. And he continued to work in neuroscience as a postdoc with mice at the University of Zurich um, since 2019. And only last week he got awarded a Swiss Ambizione Fellowship, which will allow him to start his own lab at the University of Zurich in next year, 2023. And in his spare time, he reads a lot, enjoys nature, and writes on his blog. And I shared the link to Peter's blog here in the chat. So it's a great honor having you here, and congratulations to your award that you won recently.
Thank you very much.
And if it's okay with you, Serena will ask you like a couple of interview questions first, and then the stage is yours for your presentation.
Sure.
Thank you.
Thank you.
So yes, so we're really happy to have you here and share this topic. But what we generally like to do is get, for the audience benefit, get to know you a little more about a person. So we have a couple of questions, and I want to start with: was there a time early on in your life where it really became clear that you want to get into science?
That's an interesting question. I don't think that being a scientist is a profession, but I think it's rather a state of mind. So when I was around 13 or 14, I really started to read a lot to better understand and know about the world. So I would say that I was a scientist from this point on. So in the beginning, I was rather interested in language and music, and then I moved to psychology and philosophy and ended up in neuroscience. But I think I was kind of interested in kind of science and understanding the world from this point on when I was 13 or 14. But I didn't have this picture in my mind that I would become a scientist and work in the lab. This came only very, very late.
Mm-hmm. Well, that's interesting. So at the time that you did that deep dive into science, take us from that point to where we are, and to the work that you're going to present today.
Okay, that's a long trajectory, I would say. So coming from psychology and wanting to study neuroscience because this kind of underlies psychology in some way, I thought about how to study neuroscience, and I realized for myself that to study neuroscience, you have to really understand methods and how to analyze data and to work quantitatively. That's why I studied physics for 5 years. And afterwards, I felt prepared for doing real science and neuroscience. And I looked out for labs which had the same kind of physics mindset, which means thinking about very simple experiments which could resolve very maybe tricky questions easily, instead of just making a large catalog of things which are in the world, as biologists do sometimes. So that's why I joined this lab in Basel. And at some point, I really was more interested in single cells and single neurons and how they, as some kind of organisms behave and learn about the world. And this is something I wanted to study when I joined my current lab in Zurich. However, this was changed by some circumstances because a few months before I wanted to join, a PhD student in the lab, he died from an accident. And so my PI asked me to take over this project, and in this case I couldn't refuse, and that's why I started to work on astrocytes. Actually, I wasn't so much interested in astrocytes before, but since this was due to this unfortunate event, I started to work with astrocytes, and then I became really interested in their role in information processing. Yeah, that's kind of the story. It's kind of not a straight line, to be honest.
Oh, wow, that's a complex web of events indeed. Well, at this point, we can launch into your material for the audience. We've linked your slides at the top. The original paper is in the chat, and I want to call out again the fantastic movies in the supplementary material. So encourage everyone to check that out. But at this point, Peter, the floor is yours.
Peter, just is there a way for you to make your voice a little bit louder? Maybe you could move closer to the microphone, or
Okay,
the
wait.
for us it sounds fine, but I think in the audience, usually the audio is a little bit low. People say it's a little bit low, the voice. If it's possible, it's not
Okay,
impossible.
okay. Is it better now?
Yeah, that's
Yeah,
much better.
thank you so much.
Okay,
Appreciate it.
okay. I just took the wrong microphone. Okay, let me start. So, okay, so for the first slide, the title of this talk was set by Katharina, so thanks. It's about kind of astrocytes as computational units. So I just want to define some words. So astrocytes are a specific kind of non-neuronal cells in the brain. So these are not neurons. And there's some kind of glia cells which were called like this because they glue the brain together. That was supposed to be their primary function until this has been changed more recently during the last 20 years. So computational units, computation means basically well-defined information processing. And the most important part here is unit. The unit means it's an entity. It's not about the distributed processing in the single compartments. So like in a swarm of birds or in molecules that comprise a liquid like water, but it's central processing in the soma of the astrocyte, similar to neurons. So that's the main idea of this project. So I was always interested and fascinated by neurons and information integration from dendrites to the soma. So it's obvious that such a central integration is very powerful and makes neurons the computation units of the brain. So in our paper, we found that astrocytes in the brain can be regarded as computational units of the brain as well, and that they integrate information about past events, and that this integration depends on what happens to the animal in general and what happened to this astrocyte specifically. So let's go into the slides. Let's start with slide 3. So what is known about astrocytes? This is a very coarse background slide to give an idea. So astrocytes have receptors for almost any signaling molecule, be it glutamate, lactate, noradrenaline, dopamine, anything. Therefore, it's not surprising that astrocytes respond to many different things like arousal, movement, neuronal activity, synaptic activity, and so on. At the same time, it is known that astrocytes act upon synapses, neurons, blood vessels, and so on. So they are influenced by many, many factors and they act upon many, many factors. So it's very difficult to really define their role because they have many roles and it's not clear whether they exert these roles directly or indirectly. So more recently, as Serena has mentioned just before the talk, astrocytes have recently been shown to be involved in neuron-typical computations like coding for sensory stimuli or the representation of locations. I think this is a very recent development. So this has started maybe 15 years ago, and now it has become quite common. But we can discuss this maybe in more detail after the talk, because I think this should be looked at with a very skeptical mind, because if you say that an astrocyte codes for a sensory stimulus, how do you know that this coding for the sensory stimulus is not just inherited from neurons or from arousal or from movement, which means there could be a direct effect from arousal, which seems to be an effect of representation. And if you say that something codes or represents something directly, I think that's a very bold claim which we have to support very strongly. But let's go back to the paper, to slide number 4, where I want to compare neurons and astrocytes directly. So from neurons, we have as humans, we have 10 to 20 billion in our cortex. For astrocytes, we have almost the same amount, like 5 to 15 billion. Neurons form long-distance connections and they integrate synaptic inputs, basically from the distal dendrites, which are far away from the soma, to the soma itself, the cell body. They produce a threshold output, which is called the action potential, and that's kind of the synaptic integration in neurons. Astrocytes, on the other hand, they form only local connections with other astrocytes, and activation patterns seem to be distributed within a single astrocyte. So there's an activity pattern over there and then there and there, but it does not seem to be correlated or orchestrated. So there's no obvious principle of signal integration described so far. But here I want to describe exactly what we see in hippocampal astrocytes. So the next slide, slide number 5, I want to describe the methodological part. And I think this is maybe also would become a bit clearer if you also look at the movies from the paper, because they show you how this mouse actually looks like when it is sitting under the microscope. So what we did here is we use a mouse which is head fixed under a microscope and moving on a linear treadmill. So like the treadmill that you can use in the gym, very similarly, just that it's head fixed and not fixed with the hands. At the same time, before I have implanted a window into the brain. As you can see in the middle, it's basically a cavity where we remove a tiny part of the cortex, like 3 millimeter in diameter. And instead of this cortex, we insert a metal cannula. And at the bottom of this cannula, we have a cover slip. It's kind of a window into the brain. And if we look through this window with a microscope, which we actually build ourselves, it looks like on the right-hand side. Basically, you can see the bright spots are the astrocytic cell bodies, and the blur mess around it are the fine processes, which we cannot resolve. So the activity patterns of these astrocytes are shown on the bottom in this blue and yellow color plot. On the x-axis, you see time, and on the bottom right, you see kind of a very slow process. There's 20 seconds time scale. On the y-axis, you see the different astrocytes and their time processes. As you can easily see, most of the astrocytes seem to be active simultaneously or almost simultaneously. That's the first interesting observation, which is consistent also with previous findings in cortex. On the next slide, you see, for example, how we define single astrocytes. And as you can imagine, this is not a very satisfactory analysis because you basically manually draw a region of interest around one soma of an astrocyte, and you have to kind of guess from the activity and the morphology how astrocyte looks like. So when you go back to slide 5, you can see that most of the astrocytes seem to be active at the same time, but at the same time, some astrocytes are kind of active earlier than others. And this was an interesting finding because they seem to be consistent. So for example, if you can zoom in, so if you have this on your laptop and not on your small smartphone screen, you can see that astrocyte number 85 is consistently earlier active than the rest of the astrocytes. So we wanted to understand this in a very unbiased way. And this is something I will describe in slide 7. So this is kind of a very complicated method. So I'll walk you through this, but I will promise this is also quite interesting. So what we did here on slide 7, you see a global trace of activity, which is averaged across all those astrocytes in the field of view. And then we compared this global trace with the activity of each single pixel trace. So for each pixel, we compared whether the pixel becomes active earlier or later than this global activity pattern. So we did this basically using correlation functions, which is a tool from physics actually. So I used here my physics background. And in addition, we would not be able to do this on raw movies as shown on the left, lower left, but we denoised the movies. To this end, we used an algorithm on deep self-supervised networks, which basically used the knowledge about the morphology and typical patterns to denoise the movie and get rid of this shot noise. So this is two very interesting computational methods which enabled us to analyze this data set, which has not been done before. And the result of this analysis, you can see at the bottom right, it's basically a kind of delay map. So for each pixel, we compute whether it's later or earlier active compared to the average. So in yellow, you see the pixels which are active earlier than average. In blue, the pixels which are active later than average. I hope this is kind of clear to you. And if not, don't hesitate to ask me, because I think it's very interesting method. And basically, I think that many labs have previously acquired the same data as I did here, but they didn't observe this finding because they basically didn't know how to extract this observation from the data properly. And on the next slide, slide 8, you can show the finding, how this looks like across the entire field of view. So in bright, you see these astrocytes, the fluorescence average. And on the right-hand side, you see this kind of delay map. And if you go forward to slide number 9, you see that here I manually painted some circles around each astrocyte. And this clearly shows that in the center of an astrocyte, which is the cell body, the delays are positive, while in the periphery, where the process of the astrocyte resides, delays are negative. So what does this mean? In simple words, it means that the calcium signals which we observe here propagate from the distal processes, which are far from the soma, to the soma. I think that's a very cool analysis, because this is something you cannot see if you just look at the movies. So for example, if you look at the supplementary movies from the paper, it looks really clean and so on, but these effects you cannot see just with the eye. You have to do some analysis and average across the entire recording. And I think this clearly shows the power of such methods. In slide 10, you see just another set of examples, which shows on the left in black and white the morphology of an astrocyte, and to the right the corresponding delay map, which shows that the soma is activated later compared to the distal processes. And I think this is quite a striking and interesting finding, because it shows that astrocytes don't process information just in some fine compartments, but that these signals from the distal compartments are propagated to the center, and then they activate the center and trigger some other programs. And this is something which hasn't been observed systematically before. So I think it's a very important finding to understand the role of the function of astrocytes. So it's basically a very simple analysis if you want so, but it's also, I think it's very important. On the next slide, slide 11, it becomes a bit more complicated. So here on the right-hand side, you see a couple of traces in time, and which is basically a fluorescence signal. And in yellow or orange, you see the trace which is from the periphery, and in blue, you see the trace which is from the soma. So on the left, top left, you see basically a trace where the activity propagates is small in the distal periphery and it's large in the soma, which means it propagates from distal processes to the soma. The same for most of these examples, but in the lower row, you see 3 examples of events where the activity is high in the distal processes, but not in the soma. So which means the activity from the distal processes does not propagate to the soma. So it's not the same as observed previously. And this was interesting. So basically, to sum up this finding, I saw that there's a propagation of calcium signals from the outside to the inside of the cell, but not always. Sometimes this doesn't happen. So I wanted to understand, when does this happen? When does this happen not? And so in slide 12, you see what I did to understand, to study this systematically. So basically, this slide is very crowded, but the message is very simple. So what I did is basically to systematically record a lot of other things which were going on, which is basically simultaneous neuron activity in the same brain region, the hippocampus. And I recorded the pupil diameter, which is an indicator of neuromodulation, like from noradrenaline. So even for humans, if you're aroused because you're excited or you really like this dish in front of you or something like this, your pupil also gets dilated and larger. So it's an indication of neuromodulation. Then we also recorded the mouth movement, the licking, and the paw movement, and the running. And then we systematically studied these effects and tried to understand in which behavioral conditions do these astrocytic events from the distal processes propagate to the soma, and when do they not? And we found very strikingly that if the pupil diameter is large, which means when the animal is aroused, the event propagates per 100% from the distal process to the soma. This is shown in slide 13. But we also found that if the pupil diameter is small, which means when the neuromodulation is low, and the arousal of the animal is not very high, then this propagation does not happen so often. And we could also explain the rest of the variability with cell intrinsic factors, but I won't go into this detail. I think that's a very interesting finding because it shows that the astrocytic activity propagates from the distal compartments to the soma only when the arousal is high. So to sum this up on slide 14, so together we found that astrocytes are slow indicators of past salient events. So astrocytes indicate signals from the processes to the soma. So it's very similar to neurons, which propagate the voltage from the dendrites to the soma to elicit an action potential. And similarly, we found that astrocytes integrate signals from the processes which were elicited by, for example, from neurons processing some information to the soma. We also observed that this occurs on a very long time scale. So neurons operate on a time scale of milliseconds or tens of milliseconds, whereas this slow integration in the astrocytes occurs on a long time scale of approximately 5 seconds, which is very, very long, and which is also very interesting because this is a time scale on which behavior matters. So if something happens 5 seconds ago, it is important to know about this because I can learn from this. So for example, if you ring a bell and 5 seconds later something happens, I think it's important for me to associate this distant event with this current event. So we also find that astrocytic signals reflect past events, and we find that only salient events are propagated. So to sum this up, we find that astrocytes are computational units, and with computational units, the stress is not on computational, but it's on units, as opposed to distributed and independent compartments, which has been thought previously that astrocytes would be more distributed information processing systems. And one idea that's very obvious in our eyes is that this slow integration of past salient events, which we call the conditional centripetal integration, calls for a potential role for astrocytes in neuronal plasticity. So why do we think this? So for several reasons. First, this integration occurs on a long time scale of several seconds. And we know from other studies in hippocampus that neuronal plasticity happens on this time scale in vivo. So if you look at slices, for example, you have this very short time scale plasticity, like spike timing dependent plasticity. But if you look at living animals, you see a different plasticity rules, which happens on a time scale of seconds. And astrocytes could play this role. And second evidence for this is that astrocytic signals reflect past events, according to our analysis. And third, only salient events are propagated. And this makes a lot of sense because we should only memorize and remember salient events, not the boring ones. So together this makes us believe that astrocytes could be a potential mechanism to integrate salient events and trigger neural plasticity. So on slide 15, you can see just the people who were directly or indirectly involved in this project. This is the lab of Professor Fridtjof Helmchen, University of Zurich. And with this, I'd like to thank you for listening, and I'm happy to take a lot of questions, hopefully.
Yeah, thank you so much for this wonderful presentation. And it's so impressive how you figured out which signal comes first, because that gives us so much more information about the computation. So congratulations to this amazing work. And yep, please everyone, if you have questions, flash your microphones. And so let me just go ahead, because I'm really interested in the timing of these signals. So why do you think it starts in the more distal regions? How do you think um, the network, how does it affect the network of astrocytes? Is it kind of more, because it's kind of the opposite of a neuron, like, you know, broadly speaking. So that's really interesting. So what do you think, how does it affect the network if the signaling works that way in astrocytes versus neurons?
I think the idea which is also quite accepted is that that astrocytes, they're small processes, they wrap around neural synapses. So only these tiny synapses, they feel basically when there's a glutamate transmission from a synapse. And then we assume that these tiny processes of astrocytes are activated, and only locally in these very fine processes, which we cannot resolve. And then there's basically, in our picture, a gating variable which comes from neuromodulation. And only if this gating is basically activated, this activation from the distal processes, which is wrapped around the synapses, can propagate to the larger processes. So I think this timing is just a consequence of how astrocytes react to neurons, and a consequence how they wrap around synapses with only their smallest processes.
So do you think they play kind of a more amplifier role, or just a connecting role, of connecting the network further, or more kind of, or probably also a regulator or amplifier? Like, did you see that they can kind of um, tune down the signal? Like, can they tune actively down the signal? Have you seen something like that with your imaging?
So this is very difficult to study. So I mean, if they can tune down the neuronal signals, I think how would you study this? So you can basically switch off astrocytes, but this is the same as if you basically remove all the walls from a building. Of course, it crashes down. So I think astrocytes are a fundamental part of the brain, and so they cannot be just removed and then you can look what is different. This always has some catastrophic effects. So it's not clear what effect they have, and there's
So you could use optogenetics, right? There are tools now for glia, optogenetics, and then just
yes
use like a photon or laser to activate specific, specific astrocytes, like silence them,
yes
and
the
then you
only
could see. I'm not sure if you could see maybe a difference in up or down
yes
regulation.
the only problem is that optogenetics does not work for astrocytes. So basically, optogenetics is basically you pump some ions in or outside of the cell, and it has been found that for astrocytes this does something totally different than for neurons. So it basically does not activate or inactivate the astrocyte, but for example, changes the pH or does something totally different. And it's so this is basically, if you use optogenetics to drive astrocytes, it's not clear what is going on.
And
You use uncaging of an inhibitor of astrocytes, so you don't crumble, like the whole system is not crumbling down, but just where you uncage your inhibitor.
yes, this has been done, for example, with so-called designer drugs. Maybe you're familiar with DRAGs. So these act on a slower timescale, and they basically use G protein coupled receptors to inactivate some signaling processes. But more recently, there has been found some evidence that if you use this inactivation of astrocytes, actually the calcium signal goes up. So this is very unexpected. And this just shows that we don't have really the methods yet to study this properly, to be really honest. I think there have been many studies published which used methods like this, but there are now a lot of question marks around
So
this perturbation.
could you pet astrocytes and use some, calcium um, capturing stuff in the silk?
Yes,
And
I think that's maybe one of the best methods, and it's very clean. So I'm planning to do this, but you can do this only for single astrocytes, and it's very tedious to do in vivo. You can easily do this in slices, but in slices you don't have any of these events which I'm seeing. So you have to do this in a living animal where the brain moves a lot, and then patching becomes difficult. So I have before patched in neurons of anesthetized animals, which is feasible, but once the animal really moves, the brain moves up by 10 or 15 micrometers. And that's really challenging to get a proper seal under these conditions. I think you can imagine.
yeah, thank you so much. It's really interesting, your work. It's so. And Serena, please go ahead. Thank you.
Well, thanks. Such fascinating work, and really, wonderful experimentals, bringing all that data together. I'm curious, and the time scale and the behavioral, time scale is very suggestive. So, in terms of the centripetal propagation, when it gets to the soma, do we have an indication of the information flow from there? What are the consequences?
I think that's an excellent question. I think that's also the main difficulty, because for neurons we have the axon, which is clearly the output unit of
Mm-hmm.
the neuron, and for astrocytes you don't have this. I think there are many theories what will happen, which is called the clear transmission hypothesis, and the idea is that astrocytes emit something like, for example, glutamate or lactate or D-serine
Mm-hmm.
to activate neurons. However, these molecules are difficult to study because we don't have very good sensors. So there are some labs which are developing these sensors, but we don't have yet the resolution to really pinpoint at fine processes of astrocytes and say, okay, that's released here. But that's clearly, this triggers really, our work really triggers this question very strongly. I think there's, my hunch would be that D-serine would play a big role, because D-serine has been shown in slices to be emitted from astrocytes. And D-serine is a co-agonist for NMDA receptors, which are basically the main receptors responsible for learning in neurons. So I think this could be one very promising pathway, and there's a lot of work going on which really tries to prove this. For example, I think there's some really nice work going on in the lab of Christian Henneberger in University of Bonn, which tries to show this link very directly. I think that's very tedious work because you have to exclude all confounding variables, but I think that's a very promising avenue of research.
Certainly, and yeah, in reading the consequences, well, let me just ask one last question, and we'll give more of the people on stage a chance, so I can keep talking about this for a while. The question of the syncytium, the calcium propagation through gap junctions, and your time delay methods and delay maps.
Yes.
Do you find that there's the non-local wave extended propagation from astrocyte to astrocyte within the time scale that you're looking, or are you really just seeing this centripetal integration?
That's a very, very good question. So I think this question maybe is triggered because like 20 years ago, when people started to study astrocytes using calcium imaging, they saw these waves all over the place. Especially they were using slices or cell cultures. Then they saw these waves happening all the time. And when people started to move to in vivo preparations, they didn't see these global patterns. And when I look at this delay map, as you have noticed, we don't see this kind of wave-like propagation on this time scale. And we don't see it on other time scales as well. So I believe that this propagation as a wave through gap junction is not very prominent in vivo. I think it might happen from time to time, but I think it's not the way how calcium signals are transmitted in vivo. I think the reason why there's such a global activity pattern in these astrocytes is more that the axons from neuromodulatory axons, for which are emitted from the locus coeruleus, for example, they have a very correlated activity patterns, and therefore they activate the same astrocytes all over the place in hippocampus. And that's why we think there's this coordinated activity. But we believe that there's no such thing as a global wave propagating through the tissue. I saw this a few times, but in each of these times, this was due to very bad conditions, kind of like inflammation or other extremely high laser power, which triggered kind of this non-physiological calcium wave.
Wow, that's a big result and a big question. Certainly want to come back to that. So Wisdom, I saw you flash your mic. Do you want to ask a question?
Yeah, I had a few. I tracked the Helmchem lab really closely. This preprint, I got to glance at this preprint that I think is the heart of the presentation you brought up. And there's another preprint on the dendrite work happening in the lab that couldn't be more relevant to my work. So of big interest of what's going on in your lab. But I do have a mouse on the bench. Can I ask you some like shorter questions with follow-ups? Would that be okay?
Sure.
So when you're talking about calculating the timing of the calcium signals centripetally, are you doing segmentation of the cell bodies of these cells, and then analyzing what a neuroscientist would call neural pill? I guess we'd call it astropil in this case. Or are you segmenting the processes of the cells?
Yes, very good question. So I'm doing nothing of those both. So I'm using a very unbiased approach. Basically, I'm analyzing this timing for each pixel in an unbiased way, totally unbiased. And then I'm looking at this delay for each pixel, which I compute from this correlation function method, and compare it to the morphology, which I know from average fluorescence. So I think using these manual segmentation methods that you suggested would be a bit misleading, because most of the processes of an astrocyte you cannot resolve optically. That's why I resorted to this unbiased method using pixel-wise analysis. And of course, this was only possible because I had denoised the movie before. Otherwise, for a raw movie, I would just get very noisy results. I hope this addresses your question.
Yeah. I was wondering how much overlap there was with some of the analysis going on in the dendrite work in your lab. I know they've been doing some reconstructions and fine process segmentation in neurons. But it sounds like your denoising method does try to take advantage of what is known about the morphology, if I heard you correct. And by the way, sorry, my hands are occupied, so I'm sorry if a lot of this is easily answered in your slides. But the other thing I was going to ask about was, so how much is no? It's some of this got answered in your answers to the previous questions. It sounds like these signals you suspect are synaptically driven.
Yes.
But I'm curious how much is known about the distribution, the synaptic distribution subcellularly on these cells, and also like distributions of the gap junctions. Whether do you know anything about those spatial relationships?
Yes. This is a good question. I don't know whether the gap junctions are somehow their location is correlated with the synaptic locations. This is something I don't know. I don't think so, because gap junctions are between two astrocytes, and synapses are between one astrocyte and two neurons. And the distribution of synapses, so for each astrocyte, we assume that there are something like 50 or 100,000 synapses. So I think it's all over the place, but only the fine segments, the ones which we cannot resolve, unfortunately. So all the processes from astrocytes which you can see optically with light microscopy, this is not very important stuff. The synaptic transmission of neurons is happening.
Okay, so I have two more quick ones. The other question I had, you mentioned, and again, I apologize if this is easily pulled from your preprint or the slides, but you mentioned something about simultaneous imaging. I'm assuming you're doing something like, something redshifted in the neurons or in the astrocytes.
Yes, this is something
So, sorry, go ahead. The question is, did you see any correlations between the spatial patterns in the neural activity and the spatial patterns in the astrocytic activity? Or was there any correlation there? Is it, in other words, is it? Right, go ahead.
So we recorded astrocytes and neurons from different layers, so they were spaced by 100 micrometers apart. But there's another publication which showed that there's no correlation between those. However, we found there's no correlation between the average activity of neurons and astrocytes, but if you look at the delayed effect, there's a delay of something like 4 seconds. Then if you look at this delayed correlation, there's a high a correlation between those two signals. So basically there's a neural activity and 4 seconds later there's a strong astrocytic signal. So this is, we see on average, but we don't see this on a single cell level. And we also cannot say that this is causal. We also think that more likely this has a common origin from the brainstem.
Right. Yeah. Maybe you could do like noise correlation analysis or something to try to get closer to that. So the last question I had was, forgive me, this is out of ignorance. What is known about the time course of signaling in terms of calcium versus voltage? But what I'm wondering is, how much can you determine about bidirectional signaling, like whether the signal is actually originating from the soma or distally?
Okay, that's a good question from a neurocentric perspective. So here what we measure is calcium concentration, and it is known that astrocytes are not excitable, so they don't really use voltage or active voltage channels to propagate signals. And therefore most people assume that voltage is not so important for signaling astrocytes. So more recently, so this year, there has been one paper which showed using a voltage sensors that there's something going on in distal processes. But even then, it's assumed that voltage signal is not so important. And it's not maybe not so interesting to look at voltage signals. And for, all this, I think the question arises because there's this, in neurons, there is back propagation action potential and so on. In astrocytes, this does not take place. There are no active conductances, and everything is so slow that you would be able to observe it.
Yeah. I actually asked because I didn't want to make that assumption. So it sounds like you're saying there's no evidence for active processing in these cells.
There's some evidence for some active processing in distal dendrites from this year. So this has to be confirmed, but I think it's nothing major. Yes.
Okay, thank you. Sorry for taking your time, and thanks for spending time to share your work with us.
Thanks for your questions.
So we can go, if you have a question, you can flash your mic. Barbara, I think I saw you flash your mic earlier.
I was just flashing in agreement, so I'm good. Thank you.
Okay. Are there other questions?
Question actually for Peter. Peter,
Yeah, this is John. I just have a really naive question.
Gender differences in your research?
Gender. Let's answer Barbara's question. So I think you asked about gender differences in these results.
Yeah, exactly.
So I did this work mostly with male mice, but also I think 2 or 3 female mice. And I saw basically the same thing going on in both mice. So I think this is something which I think this is a very first order effect, which is very obvious. So I think I'm not surprised that it's the same for female and male mice. But I could imagine that there's some minor differences when you look more closely. For example, it is very obvious that male mice behave very differently from female mice. They're usually more aggressive, they're more quicker to learn something, but also make more mistakes. And they're a bit more difficult to handle and less anxious. So there are many differences on the behavior level which are obvious. I think this would surely reflect somehow indirectly in the astrocytic activity as well. But I don't think that astrocytic activity drives these differences. Thank you.
Thank you, Peter. Appreciate it.
John, do you want to?
Yeah, did I understand you correctly? You said there's between 50 and 100,000 synapses per astrocyte. Did I hear you correctly?
Yes.
So that I'm not a neuroscientist, but that raises some really interesting computational questions about staging of processing, simultaneity of grokking 50,000 potential inputs or 100,000 potential inputs into a single cell. Are there any early hypotheses about how a single cell, obviously not all 50,000 synapses are firing simultaneously, yes, but clearly the capacity for a very diverse set of signals arriving either near simultaneously or in some sequence that means something different than a different sequence. Are there models for how to reduce the complexity of that massive input network into a single cell that have been tractable experimentally?
I think not. I think but I can tell you what I think about this problem. I think it's a really good question, which way too few people in the astrocyte field are asking themselves, because I think it's an obvious question, and it's you ask yourself what could this be good for? And one idea I had was that maybe so when a synapse is active, it basically contributes indirectly to this activation of the soma. And it could be that this synapse is thereby, by its own activity, kind of tagged. And once the soma is activated 5 seconds later and emits some, for example, D-serine, which potentiates synapses, this tagged synapse could be activated. So it's basically, this could be used to, that there's no, if we look from the soma activation, it does not propagate to all the synapses, but only affects those which had been active before. This is one way I could imagine how to deal with this huge complexity and diversity of inputs by tagging them by activity. So this is just one approach to think about it.
Yeah, I mean, you could think there are two sort of polar strategies across a spectrum of strategies. One is in a particular localization of the astrocyte, there could be some edge computing across the different receptor types, or there could be a central summation, and at the other end would be a central summation of all of it relatively simultaneously, and somewhere in between those two is that there's some, cross receptor type edge computing, anatomically localized, not anatomically, spatially localized within the cell. I mean, there's just so many different ways that problem could be solved for both the variable types of receptors, the variable location of those receptors on the cell, the localization within the cell. I mean, the number of strategies that could have evolved to manage that information, pretty diverse and complicated, and seems like it could have a significant reveal in terms of neuromorphic computing and biomimicry in the digital space. Just curious if you have any thoughts about that.
Yes, you've brought up a very interesting point that you mentioned two different strategies which the astrocyte could use to compute, basically. And I think it's, or even more than two. And I think the answer is that it will be just more than one strategy. I think probably the astrocyte is using both strategies because from my experience, biology is very messy. It's not engineered, and it uses just any kind of complexity to do whatever it needs to do. So I think I could well imagine that it just uses both at the same time, maybe even somehow mixed. So I think I'm not sure how to implement such messiness in neuromorphic engineering devices. I think that's a very good question. Yeah.
It'd be interesting to do some reverse biomimicry and look at how these problems are being solved in the IoT, Internet of Things.
Okay.
It'd be fascinating to see how people are thinking about the equivalent problem in sensor integration across a huge network where there's localization of multiple different signals available, but there's aggregation centrally as well. So it'd be really interesting to see the digital analog of that and then use that as a probe into the biologic analog of that.
That's a very interesting thought. Oh, thanks for sharing.
Rajarshi?
Thank you. So I'm just going to try and understand it out loud, and please correct me if I'm wrong. So, the brain, in an effort to crunch a lot of analog signals into more efficient processing, it sort of makes shortcut pathways for repeated signals. And I think of it like, if I'm trying to think of how to make that efficient, instead of trying to store individual timestamp data points of your time series of signals, you just store like a record of all the frequencies. So it tells you like I've got 10 signals of this approximate intensity over the lifetime of that pathway. So I'm just trying to understand, is that like the role of astrocytes to get these analog input signals and sort of keep track of incoming signals, but also inform neurons that are doing the calculations of what the average value is and what the instantaneous value is relative to those average values, while also updating this record?
That's a very, very engineering perspective. It's funny to hear it put in these words. Let me think about it. So your idea is that basically astrocytes could be described as integrating past events and then basically using this average across time or smoothed signal and feed it back to neurons. I think this could be one role for astrocytes. I think it's but I guess it's not, maybe not the entire thing. I think there's more to it because astrocytes are so closely involved in the regulation of synapses and neuron plasticity. I think there's more to it. Just computing the mean or some other statistics over the past events, I think that would be a bit too simplistic. And I think you don't need 15 billion cells in the cortex just to do this. I think that's something you would do with a neuromodulation signal, which is globally emitted, and there are some circuits for this purpose.
Oh, thank you. And what? Sorry, sorry. If it's a follow-up question, please go ahead, John. I can wait.
No, go ahead with your follow-up because mine is a follow-up to my question. So go ahead and complete yours.
What's the role of calcium and in the astrocytes, and what calcification I think it's called? What's the role?
That's a very good question. I think it's not fully understood. So people have been convinced that calcium, for example, in the soma, triggers some molecular cascades, which lead to the release of some neurotransmitters like D-serine. So that's why people assume that calcium is a very important signaling molecule. Also for other cells like neurons or other cells like heart cells or muscle cells, calcium is very important. So that's why people thought it's the central signaling molecule. But more recently, in the last 1 or 2 or 5 years, there has been also some processes described in astrocytes which are important, which do not depend on the calcium concentration. So I think there is more to it than this, but in general, I would say that it's the most important signaling molecule to activate some molecular signal and cascades.
And just one last quick one. You mentioned that voltages were part of the signaling, but like a minor part of the signaling. So what is the major? How is the signal transmitted? What if not voltages?
It's transmitted not by. I think it's transmitted by molecules which are, for example, which are released by vesicles into the extracellular space. That's the main route of transmission.
And are these the same or similar neurotransmitters, neurotransmitter molecules that we expect in, for example, neural synapses?
Not only, it's some of those, yes, but there are additional ones like lactate or D-serine or some ATP. These are much more diverse than for neurons. For neurons, it's usually one transmitter per neuron, which is effective, and for astrocytes, it's a huge variety.
Okay, and just one last quick one. So there was an experiment where you take a plant and you drop it, say a touch me not, and like make it fall like a height of 2 feet, and you suppose you do that every hour. Say the plant visibly curls up when it's being dropped from a height. And then the first if you do it every hour, then after the first few hours, it starts to curl up even before you've dropped it because it's expecting that input and expecting to do a defense. And then after a couple of times of doing this and realizing that it's not getting hurt, it stops curling up at all anymore. So would you say that this mechanism of how it figures things out is similar to do plants have astrocytes, or what's the sort of equivalent that helps them do this function?
Oh, that's a very I would have to look into this. I really don't know, to be honest. I think that's an interesting observation. I would have to look it up. If you're really interested, you can send me a link to this observation, and I can look at it and write you back. But I think without knowing much, I cannot really tell. Sorry.
But is it exclusive to mammal brains, for example, or do you find astrocytic things in even simpler neural nervous systems?
You see it also in simpler nervous system. Even in insects, you have some precursors of astrocytes which look differently, but they have some similar functions. So this is to some extent conserved across species, but only I think since reptiles they have this astrocytic morphology with this star-like shape.
Okay, thank you so much for taking my questions.
You're welcome.
Yeah, I have another naive question. So, to what extent do you have any sort of proxy metric for computation itself? And is it possible that computation is detectable from a energy consumption or redox kind of chemical transaction? And if you have the ability to look at the energy expended in computation versus transmission, would that be an interesting probe into whether, to what not whether, but to what extent computation is done at the edge versus computation is done more centrally or at every step along the way? Is energy expenditure something that is a potential proxy for disambiguating those options?
That's something I haven't thought about so far. It's an interesting point. Just one clarification. So the computation that I'm observing here in this study is basically very simple. It's the computation, it's a signal transmission from the distal process to the soma, and which is transmitted conditionally only when arousal is high. This is the computation I'm observing here. I'm not observing something very the computation of input signals or location or something like this. But to your question, I think it's very difficult because energy consumption in biological tissue can be measured using ATP as a unit, because ATP
Right.
is.
Yeah,
But
that's what I was assuming you would look
yeah.
at. Yeah.
But at the same, there are some. I think some groups have now developed some ATP sensors. They are not very sensitive, so it's not very nice. And another problem is that astrocytes also use ATP as signaling molecule. So just to mess up everything. So I think it's very difficult to quantify. And then you have to count for something like volume to surface to volume ratio to account for whether processing happens on membranes or in the volume. I think that's very, very tricky. And for these astrocytic processes, we cannot resolve them. Even with super resolution microscopy, they are too tiny. So we cannot really study this in vivo right now, unfortunately. But it's a good question. I just don't know how to address it.
Thanks.
Thanks for the question.
So I'm curious. I mean, your data presents the the multi-second centripetal propagation quite well. In some of my readings, I've come across some faster processes, and I'm curious about. Well, let me pose it as if in the role of de-searing release and plasticity, the with 50 to 100,000 neurons, I'm imagining that the origin of the distal stimuli are forgotten by the time that propagates to the soma. But in the sense that de-searing or other glial transmitters may affect synaptic plasticity at the distal processes. Is there a faster time process that could be occurring that you could discuss? I've also heard or read that astrocytes are involved in theta rhythm, which would be much faster than the timescale that you're looking at. Any comments on any of the more local or feedback coupled loops to the synapses at a faster timescale?
Yes, I think there might be so about the slower time scale. I think if it goes over the soma, I think it will be a slower time scale because the soma activation happens through diffusion, and diffusion is always slow.
Mm-hmm.
And I think this you mentioned that you assume that the memory about these processes which happened before is forgotten at the synapses. I think this must not be the case. Doesn't have to be the case. I think there are models of synaptic tagging which could make the synapses memorize what happened a few seconds before. I think there's some evidence from neuronal recordings, for example, in there's from I can type in the chat from 4 years ago, actually.
So would it then be there's sort of another question in terms of if the antecedent, so you mentioned that there isn't clear causality, but presuming that the synaptic activity initiated, or it was at least correlated with the origin of the distal calcium waves toward the soma.
Yes.
In terms of any feedback that may occur, would there be a mechanism to jump to synapses that weren't part of that original stimulus, but happened to be in that right place at right time, that they got the benefit of the association between arousal driven by the norepinephrine and the calcium integrated signal, for that region and time? Now, in other words, is there a way for activity of synapses in one area to affect through these astrocytic processes synapses of other neurons that are in the area within the tiled pattern or the region of the astrocyte that weren't necessarily active at the time?
That's an interesting question. I think this could be the case. So this is something is not covered by our experimental data, but I think this could be the case because there's some local activation of some internal cascades, G protein coupled receptors to noradrenaline, and these basically release internally in stored calcium, and so there's some local amplification loops where signals which are received from synapses, for example, could be amplified using this neuromodulation or noradrenaline. And this could indeed also affect these local synapses spatially. The reason why I think I'm hesitant to follow up on this is because in hippocampus it is known that neurons are distributed basically randomly. So the tuning of neurons which are proximate, close to each other, they are not co-tuned a lot. So I think it would be a good, following up on your question, it would be interesting to see whether synapses which are close are actually correlated, because synapses are not distributed in the same manner as neurons. So I think it would be an interesting study, but however, I think it's very challenging to study synapses.
Mm-hmm. And in terms of the heterogeneity of astrocytes, I mean, you're studying CA1 area of the hippocampus,
Yes.
where you had 3 layers, I believe. But the astrocytes are widely distributed throughout the brain.
Yes.
Do you think these? Is there an indication that their roles may be different in say in the cortex regions, for example?
So my hunch, I don't know for sure. So if I look at calcium recordings from cortex, I mostly see very similar things. So I think very similar things are going on. However, there have been some reports that, for example, in prefrontal cortex, astrocytic activity looks a bit different. It's not these global patterns, but very more distributed ones. So there could be some different things going on, and it holds especially true for subcortical areas, which is, for example, the brainstem, the spinal cord, and so on. I think or the cerebellum. I think there are different things could be going on. I think all of them, most of them, are similarly modulated by this neuromodulation, but I think how they interact with neurons could be very different. That's clear, and I think there's a huge open question mark about this heterogeneity.
Because, some of the really interesting work that involved the, syncytium and propagation was regionally focused, and I don't believe it was all sliced. It's a curious question and a big one if the importance of, multi-astrocyte syncytium type dynamics is a regional and computationally specific type of role that isn't present in the hippocampus. That would be kind of a fascinating thing.
Yes, I think it will probably is very similar in some regions of cortex, but I think it's without this cortical layers in, for example, striatum, I think maybe there's something totally different going on. I think it's, yeah, it's a good point.
So are there other questions from the audience? Listen?
I was just a quick question. I was wondering about the details on how you expressed the GCamp sensor. Was it targeted towards specific cells, or was there an effort for sparsity? And just curious in terms of, and if that is the case, I'm just curious how many cells you were looking at relative to, how many astrocytes would be there. And then the last question is, did you see a variety of responses, and if so, how many of, percentage-wise, how many of the cells supported the main result of the paper?
And these are good questions. So we basically try to use viruses to express GCamp 6S in all astrocytes using the GFAP promoter. So it is known that this expresses also in some neurons, but we could identify those very easily based on their morphology and their activity. And when we looked at the GFAP staining using immunohistochemistry, we saw that basically all of the astrocytes in the field of view were labeled. So about the second question about the heterogeneity of responses. So we saw that some astrocytes indeed did not show this centripetal propagation that we observed in most astrocytes. And we haven't saw this kind of outlier astrocytes we saw in some of our field of views. In others, it was not really clear because if you don't have the soma of the astrocyte in the imaging plane, it's unclear whether you're really imaging an astrocyte or which part of an astrocyte you're imaging. But in a few cases, we could really clearly say, okay, this is an astrocyte, and this astrocyte doesn't show this typical centripetal propagation. To put a number on it, I haven't done. I would say maybe 10 or 5% of astrocytes don't show at all this dominant pattern that we observe.
Okay, thanks. Thank you.
Welcome.
Other questions from the audience?
Yeah.
Please.
Oh, go ahead.
Oh, thank you. So you mentioned that we have troubles with resolution of these signals. Just in your opinion or perspective, what might be the sort of scale that we should be looking at even if it seems ridiculous right now. But how narrow should our resolution be ideally to really capture all this information?
Okay, that's a good point. I think it's these fine processes. I think they're on the scale of maybe 500 nanometers or 1 micrometer. So it's very wait. Let me think about it. I think it's maybe 200 or 300 nanometers if I'm right, but I might be wrong. I'm just remember that it's below the resolution limit of light and light, which is kind of like 400 nanometers or so, and I think it's below this this region. And how can we resolve this? I think I don't see a way forward. So I'm basically trained with microscopy. So I think I have an overview of the available methods, and I don't see how this can be achieved. Of course, we have electron microscopy, which you can use on fixed tissue, and this can give you some information. And I hope to see some analysis of astrocytes using volumetric electron microscopy data sets in the future. But for functional analysis, I'm not so sure how to go forward. I think that's an open question.
And I understand that your method uses a video, and I'm wondering, in terms of temporal resolution, I'm assuming because you have like a limited number of frames per second, you can only achieve so much temporal resolution. But in both in your case and in the ideal theoretical case, what might be the temporal resolution you'd want to get like all the information out of this?
I think the temporal resolution we have, which is necessary. So there have been some studies which have tried to go to the limit. And in this case, we have a temporal resolution of 30 Hertz. And we observe any processes that we can observe based on our spatial resolution are much slower. So on a time scale of, I think the fastest are maybe 200, 300 milliseconds, which is much longer than our temporal resolution. So on a temporal resolution, we are totally fine.
The
Okay,
rate limiting thing here is the GCamp sensor.
I don't think so, to be honest. I think the rate limiting factor is really the processing by diffusion in the astrocytes. I think for some tiny events in the distal processes, the GCamp might be the limiting factor, but there are some other studies which show that it's not really the case.
What you're saying is you think the GCamp sensor is fast enough, but there's nothing optically or imaging wise you can do to go faster than the GCamp signal, right?
I think we could do, for example, GCamp 8, which has a much faster rise time. So we haven't done this. This could be done, but I expect.
But if you're saying that the calcium signal is slower in the cell, then there's no need to.
Yes.
Right? Yeah.
Yes. I think there might be some small processes which are activated very quickly, but then these are very difficult to resolve. And then to be sure that this is really a signal, not just noise, it's difficult. Yeah.
Yeah, I have a question. So we had a guest speaker here like weeks ago, Moritz Armbruster. I don't know if you know his paper, Neural Activity Drives Pathway-Specific Depolarization of Peripheral Astrocyte Processes. And so his work is mostly slice and culture, but he saw that these distal processes were mostly driven, by potassium, and that by action potential mediated presynaptic potassium efflux and electrogenic glutamate transporters. Yeah, again, this is easy to do in a slice, but in vivo probably not so easy. But do you know about this paper? And because this could basically explain, the activity that you see, and that it starts kind of in this distal regions, and it's potassium driven mostly.
Yes, I know this paper. I have to say, I think it's a very methods-based paper because it's also very strongly based on this voltage imaging method. So about this, you summed it up that the main effect that the neurons exert on these distal compartments are these potassium release. To be honest, I cannot really judge whether this is because true or whether other models are true, because there are other models which claim that molecules emitted from or ions emitted from axons are responsible for triggering astrocytic activations. But I think it could be very, very true. So I don't remember the details of the paper, but yes, I think this could be a link, one of the missing links to explain why the distal processes are activated first, or how they're activated first. I think it's clear that it's.
Go ahead.
Yes, I think it's clear that it's synaptic activity, but
Would
it's.
you consider using this encoded voltage
Katerina, I think you're cutting out. You're a little matrixy. But I remember that work too, and one of the takeaways was that even the release of glutamate into the synapse, there was a voltage of about 20 millivolts at the astrocyte in the tripartite synapse, and the voltage sensors led to an influx of calcium, and I'm wondering if the mechanism there is just through the release, the voltage difference, and the influx of calcium, and if the calcium wave could be initiated through that mechanism.
Yes, I think that's also okay. Now you said it's glutamate. Yeah, it makes more sense. Yeah, I think it's probably true. I think it's initiating the wave, but I think it also requires the neuromodulation by noradrenaline, because this also triggers some pathways which amplify this initial calcium triggered by the maybe triggered by the depolarization. Yeah.
Well, in that paper, it was more potassium focused, so you could probably block voltage-dependent potassium channels locally, and see if something changes.
Okay, I think it's yeah, I think that's always difficult because I think that's a good point. I think only technically there's a challenge because voltage-dependent potassium channels are everywhere without them don't have action potentials or no proper action potentials. So if you apply it just systemically, you would just block it out, would make a mess of the brain. So you would have to basically patch a neuron and apply these potassium channel blockers locally to this very neuron, which is very challenging if you want to at the same time have a behavior of the animal recorded. I think that's maybe the dream experiment to really nail this down, but that sounds really, really challenging.
Yeah, is there a way to do uncaging with UV light in vivo in some form?
Yes, you can do uncaging, but again, the spatial resolution of uncaging will be determined by light, and you cannot say, okay, I want uncaging happening in this synapse. So you don't know whether you're really doing this on a synapse or on some neural dendrites or some other things. So this is
Well, in slice you can, but I'm not sure if you can do in vivo. There is this holographic way of doing this, but I'm not sure how well it works in vivo with things moving around. It would have to be in anesthetized animals.
I think you could do this also with a living animal. However, for holographic activation or uncaging, you still have the resolution limitation will be something like 2 or 3 or 4 micron in micrometers, which encompasses hundreds or thousands of synapses. So I think it's very
Well,
difficult to.
I did that, and my supervisor at Stony Brook did local uncaging just on specific synapses, but of uncaging of glutamate. You can do that. I can send you papers about it just in slice, not in vivo. So in vivo, I'm not sure because you have a lot of stuff you have to get through. But yeah, people have done it to measure like upstates and stuff like that. You can very precisely on a synapse, and then 5 synapses and so on. Uncaged.
Sounds interesting. Okay. Yeah, I would be happy about to see the paper on this.
This might be a bit of a basic question, but is glutamate, for example, used in specific contexts, or is it like used as just a signal level, or is it only for particular signals that glutamate is used versus another neurotransmitter? And if it's like used for a particular case, could you give, for example, why would you use glutamate?
So glutamate is used mostly by neurons as a neurotransmitter, which are excitatory, and inhibitory neurons in the mammalian brain use other like GABA as neurotransmitters. And in addition, also astrocytes use glutamate as a transmitter, basically. However, in this case, the role is not so clearly defined. I'm not sure whether this answers your question.
Oh, it does. Thank you. But is this relation like mutually exclusive? Is glutamate the only thing that's used to excite? And or does it have other uses as well?
That's a good question. I think it's used to excite postsynaptically or drive glutamatergic receptors, but in addition, it also is involved in long-term memory because they are not only the simple glutamate receptors, but there are also the so-called NMDA receptors, which are also glutamate receptors, but they need some additional depolarization from the intracellular side. And once this happens together with external glutamate, then they can trigger some potentiation, which means a strengthening of the synapse. So there's a secondary role of glutamate, yeah.
And the other way around, are there other molecules that are used for excitation besides glutamate?
Yes. For example, if you want to activate some muscles, there is acetylcholine, which is used. It's just a different neurotransmitter. And in addition, of course, there are some other neuromodulators like dopamine or noradrenaline or serotonin. And these are not really the same kind of exciting neurotransmitters, but they also contribute to the excitation or inhibition balance of the network.
Wow, that sounds incredibly complex. I think my respect for everyone working in neuroscience just went 5x. Thank you so much.
Thank you.
Go ahead, Karen.
Oh no, I just wanted to ask if people had more questions because we've been going for 1 hour and a half. So Peter is probably late. We're in Europe, so I just wanted to make sure we don't, overstretch your patience.
No problem.
Well, okay. Let me get a final one in. In terms of the balance of excitatory or inhibitory synapses, do we find a mix of them within the region of an astrocyte, implying that it could be either an excitatory or inhibitory initiation of the signaling that ultimately gets integrated, and sort of a magnitude of activity as opposed to the sign? or are they fairly regionally specific?
So you're asking whether the astrocytes are specific for interneurons or excitatory
For
neurons?
glutamatergic or GABAergic, either excitatory or inhibitory synapse.
That's a very good question. I haven't thought about this. I think I know of reports how astrocytes respond to inhibitory and excitatory molecules, and I would assume that normal astrocytes would respond to both, because astrocytes basically tile the entire space in 3D, so they don't overlap with their territories. Therefore, I would assume that if there's a synapse, inhibitory synapse, it would have to be covered by the same astrocyte, which also covers an excitatory synapse. But I don't know for sure because I haven't thought about this before. Good question, yeah.
I would also point out, I that's a really interesting question, Serena. I haven't thought about that either. But even if it were the case that they were perfectly selective for excitatory synapses, that doesn't mean that the activity at an excitatory synapse couldn't start a signaling cascade that ends up affecting inhibitory synapses, like, maybe perhaps locally along a dendrite or something like this. So even if you did have precise selective targeting, it doesn't mean that only the excitatory synapses are going to be affected.
Well, certainly, and I think it is mixed if it's simply a spatial domain. And what's interesting about integrating the signal over time is what you would be amplifying is the particular balance of excitatory and inhibitory activity that occurred at that time, which is its own kind of, which is a more complex signal to encode in essence, as it wasn't just the particular pathways of excitatory or inhibitory signaling, but it was the balance of everything that converged on that region and associated with arousal at the time that becomes the salient event that gets remembered.
That's a very good point. Yeah, I think it would be very interesting to see how single astrocytes reacts differentially to inhibitory and excitatory neurotransmitters. How this is integrated, I think your summary makes a lot of sense to me.
Any more questions from the audience? If not, I really want to thank you for sharing this work for us. It's a fascinating topic, and it's just an amazing presentation and analysis of the data, and very illuminating. So thank you very much.
Thank you for your very good questions. I was really positively surprised by all these creative and interesting questions. So it was a really pleasure to be here. Thanks a lot for the
Yeah,
organizers,
thank you.
especially.
Oh, thank you so much, Peter. It was really a wonderful talk and such an interesting discussion. It was really, really amazing. And I wish you all the best for your lab and that you get a lot of funding because it's very important work to better learn about the role of astrocytes, and you develop this interesting techniques. Yeah, I wish you all the best, or we all wish you all the best for your lab, and congratulations again. And thank you so much. And if you maybe have some updates next year with your new lab, please come back and share your insights with us. That would be amazing. Thank you.
Thank you very much.
Okay, and thank you everyone for coming, for asking great questions. If you like discussions like this, follow the club. We will have more talks this week. We will have Dr. Pascali talking about 3D printed biomimetic artificial muscles. Then we have Dr. Gagnou talking about mutations that protect from cognitive decline. And Dr. Oliveira Maia, He's a director of the Chapelimu psychiatry department in Lisbon, and a friend and colleague of mine. He will talk about goal-directed and habitual actions and neuroscience studies. And then on Friday, we'll have Dr. Michael Levine, the lab that developed the xenobots and other really interesting recent research. He will give. He was here before, and he will give us new updates about what's happening in his lab about bioelectricity, basal cognition, and more. And it's like even unpublished work. So if you want to hear and get updates what he's up to in his lab, join us on Friday, and yeah, we will have interesting discussions. And again, thank you so much, Peter. This was really an outstanding discussion and talk. So we really appreciate it, and good luck for everything.
Thank you.
Okay, we'll close the room in 3, 2, 1. Bye, everyone. Thank you.
Thanks, everyone.